Namaskar. Today we'll study the G protein coupled receptor and its different pathways and second messengers. So G protein coupled receptor, also called as 7 transmembrane receptor, because it is located on the cell membrane and it is helical structures surrounding the cell membrane. So it is 7 transmembrane receptor and looks like a snake, so it is also called as serpentine receptor, and it is called G protein coupled. G stands for guanine nucleotide. Guanine nucleotide can be either GDP or GTP. Guanine nucleotide binding protein. This protein has 3 subunits, alpha, beta and gamma, and guanine nucleotide is bound to the alpha subunit. So alpha GDP, when it is bound to alpha GDP, it is inactive or in resting state, and when it is active it becomes alpha GTP. So alpha GTP and beta gamma are separated. Two parts are there in its active form, and then this alpha GTP and beta gamma can bind to any effector to produce their effects, and the effector can be either enzyme or ion channel. Two enzymes are there, adenylate cyclase, phospholipase. Two ion channels are there, potassium and calcium. And adenylate cyclase produces a cyclic AMP from ATP. Phospholipase, different phospholipases are there. Phospholipase C mainly produces two second messengers, IP3 and DAG, inositol triphosphate and diacyl glycerol from PIP2. So this is cyclic AMP and IP3 DAG are the second messengers in G protein coupled receptors, and similarly these ion channels, potassium and calcium ion channels are there, and and we can see adenylate cyclase produces cyclic AMP from ATP, and phospholipase C, by acting here, can produce inositol 1,4,5 triphosphate from phosphatidylinositol diphosphate and diacyl glycerol DAG. This part is DAG, this is IP3 by the action of phospholipase C, and here phospholipase A2 can act to produce arachidonic acid. So if you see the G protein coupled receptor, so in its resting state alpha GDP is there. When the agonist binds, GDP GTP exchange occurs. Alpha GTP will now become active and get separated from the receptor as well as beta gamma complex. So now alpha GTP and beta gamma complex, these two can move freely inside the cell membrane. They are freely diffusible in the cell membrane and have high specificity. They will bind to specific effectors on the cell membrane, and these effectors can be either enzymes or ion channels. Then after binding to the specific effector, they will produce their cellular effects. Then there will be activation of the GTPase enzyme by a regulator of G protein signaling. We call it as RGS protein, regulator of G protein signaling. Then that will activate GTPase. So GTPase will hydrolyze GTP, a phosphate is released, GTP becomes GDP, and when it becomes GDP, alpha GDP, it will get separated from the effector, and simultaneously there will be separation of beta gamma from its specific effector, and alpha GTP and beta gamma will return back to the receptor to its resting state. So this is how the cycle of G protein coupled receptor functions. Now these G protein coupled receptors have different families, family 1, 2, 3 and 4. Family 1 has two subfamilies, GS and G olfactory. So GS has subunits alpha S, G olfactory has subunit alpha olfactory. Alpha S activates adenyl cyclase, opens calcium. Alpha olfactory activates adenyl cyclase. Similarly, the second family consists of GI, GO and GT. GI consists of alpha I, which inhibits adenyl cyclase. Beta gamma opens the potassium channel. GO alpha O activates phospholipase C. Beta gamma opens potassium, close potassium. GT subunit is alpha T activates cyclic GMP phosphodiesterase. Then GQ, the family 3, subfamily GQ, subunit alpha Q activates phospholipase C beta, and family 4, G12 or 13, alpha 12 or 13 causes the sodium potassium exchange, sodium hydrogen exchange. So these are the functions, and so mainly this adenylate cyclase and phospholipase C, these are the two targets, and accordingly there is second messenger cyclic MP IP3 DAG, and there can be opening or closing of this potassium and calcium channel by G protein coupled receptor. So what happens when the agonist binds to the G protein coupled receptor? There will be GDP-GTP exchange, and alpha GTP or beta gamma will bind to their specific effectors to produce the second messengers, and those second messengers like cyclic AMP, IP3, DAG, they produce they show their effects, cellular effects. Then there will be activation of GTPase, hydrolysis of GTP bound to alpha. Alpha GTP will then return back to its resting state. So how the cyclic AMP and IP3 DAG functions? Let us see one example. IP3, this is a GQ type of receptor where phospholipase C is the effector. IP3 acts on the sarcoplasmic reticulum to release calcium. Calcium binds to a cytosolic protein calmodulin. Calcium calmodulin complex activates other protein kinases and produce cellular effects. Similarly, diacylglycerol in presence of calcium and phosphatidylserine activates protein kinase C, which activates by phosphorylating other proteins to produce cellular function. Similarly, in case of GS type, following adenyl cyclic AMP pathway, this is one example where alpha SGTP will bind to the adenyl cyclase, cyclic AMP will be produced. Cyclic AMP activates the protein kinase A, which produces the cellular function. Protein kinase A has two regulatory subunits and two catalytic subunits. When the cyclic AMP binds to the regulatory subunits, catalytic subunits are released. They are the active form of the protein kinase A, which will produce the cellular functions. So in this way, G protein coupled receptor shows its function. Thank you.